So for the multi-omics integration, the basic idea is that if you have the technology available to capture multiple omics, for example, RNA and protein coming from the same sample and from the exactly same cells, then we can either analyze them individually, but we can also combine the information from both sources. In this case, we are going to use the SID-Assist technology. This technology is also coming from 10x Genomics. And here, basically, you are providing the same type of sample as you would do in the Visium technology. So you can, again, capture the tissue morphology using H&E staining or immunofluorescence. This is only for capturing the tissue structure. Now, on the other hand, you will capture from this sample the whole transcriptome, so you will have RNA, but you can also do immunofluorescence, so you will use proteins that attach or recognize surface proteins in the cells. So here, unfortunately, right now, the catalog for capturing the surface proteins is not too big. Right now, the availability is only to capture around 31 proteins on the surface, but it's still giving good results so far. This has been developed right now for human tissue, if I remember well. I don't remember if they already have um, uh, what's the name? Like immune detection for mouse. I'm not sure if they have already for mouse, but you can keep an eye and look for more details in the website of 10x Genomics. So for this exercise, we are going to use a sample coming from human. So, they have for human 35 antibodies, not 31. So they have 35 antibodies, and then regarding the RNA, you have whole transcriptome. So for this example, we are going to use the human tonsil dataset. This is a publicly available dataset in 10x Genomics. You can look for more information about how they process this sample in the website. So following this link here. And you can download the raw counts directly from 10x Genomics website using this code. The downloaded files will look very similar to the structure of the Visium data. So basically, you will get two files, one with the expression matrix. Here you can see that, in fact, the files have a very similar name. So we have the raw feature VC matrix. They also provided the filtered feature matrix, but in this case, we want to do the processing of the sample. So we are going to use the raw counts. And this matrix contains both the information from the RNA and the protein in the same matrix, but they have some labels to differentiate what values are coming from the RNA and which values come from the protein acquisition. And then we need also the spatial information, which is basically a matrix very similar again to the Visium. Here we will have the cell IDs and then the X and Y coordinates. So once that you unzip these files, you will get the organization in a very similar arrangement as before. And then you can create a Giotto object also following kind of the same structure. So basically you need to load the Giotto object. Then you can set the instructions such as where you want to store your results, the Python path that you can leave just as null, and it will the Giotto functions will do the automatic detection of your Python path. Then you can specify whether you want to save or show your plots. And you have to indicate where is your data located. So basically, you want to provide the path to the folder that contains both the raw the raw expression matrix and the spatial folder. So they will look. Let me show you just how they look in general. So basically, once that you perform the the unzip, you will have these two folders. In this one, we have the three files that we already know, the barcodes, features, the matrix, and then we have the low resolution image as before and the high resolution image. So as you can see, this structure looks very similar to what we already know from Visium. Now, the function create Visium object is already adapted to be able to detect when we have only one feature in the expression matrix, or detect that we have multiple omics in the expression matrix. So in this case, you don't need to specify the feature type. It will automatically detect that you have both RNA and protein. And it will create two separate feature types in your Giotto object. So this convenience function is very, very useful because you don't need to handle manually the reading of the files in the expression folder, and it will automatically create a Giotto object. Now, from here, I will read my object directly from my folder. So if we go to our studio, we need to load Giotto. Then I'm going to load the Giotto object. You have available this Giotto object in the files from Zenodo. So you can either run the code with me, or you can already download the Giotto object, which has some pre-processing steps. So if we print the contents of this Giotto object, I already performed some steps here. But what I want to point to you is that under the expression slot, we have two feature types, which is the RNA and protein. So this comes from the automatic detection of the convenience function. And when you just created the Giotto object, you will see this raw expression matrix. You won't see the normalized and scale yet if you run the code from the instructions. Now, when we create, I think I still have enough time, so probably I will run the code with you. Let me go to the beginning. Okay, so we have the results folder, the Python path, instructions, and then we create the Giotto object. Okay. So I will just print again the contents of this Giotto object to show you how it looks like without the other processing steps. So here we observe that we have RNA and protein. We only have the raw counts right now. And we have the image. Right now we are reading the low resolution image, but you can change this into the high resolution one in case you want to do that. Then in the same way as with the Visium dataset, we need to first filter those cells or spots that are not covering or are not matching with the tissue area. So if we go to the metadata of this object, we can observe that we again have this column indicating in tissue where 0 is not over the tissue, 1 is under tissue. So basically we want to remove those cells that are not really over the tissue. So right now it looks like this. So you can observe that we still have the spots not matching with the tissue. So we are going to do the same as before. We want to get from the metadata that column and only keep the cells that are covering or matching with the tissue. We are going to subset the Giotto object. And now if we take a look again at the metadata of this object, then we only have cells that are overlapping with the tissue. Now, before doing the multi-omics integration, we need to process the individual omics. So in this case, we need to do the processing of the RNA modality, and then we are going to process the protein modality, and then we are going to integrate the results. So we need to perform the first steps until the dimension reduction step. Those are the minimal requirements for doing the multi-omics integration. In this case, I'm going to also go until the step of calculating the latent clusters because I want to do the comparison between the results of the clustering doing considering only the individual modalities, only the RNA or only the protein. And then I want to show you how it looks like with the integration. But in case you are creating your own pipeline, you don't need to go to the clustering necessarily. At least you need to perform the steps until the dimension reduction or the PCA calculation. So I'm going to perform the filtering of the RNA. I already know that these parameters work well for these data sets. So I'm setting the minimal number of cells with a certain feature equals to 50, and then the minimal number of features per cell equals to 1,000. Then I'm going to normalize the RNA counts. So here, if we print right now the object again, you can see that for the RNA modality or for the RNA omics, we have already the raw, normalized, and scale expression matrices, but for protein, we don't have additional matrices. We just have the raw one. Then we are going to calculate the statistics. I will do a calculation of the highly variable features. Now I'm doing the highly variable features here for RNA because I have information at the whole transcriptome level. But if you face the situation where your technology is not the whole transcriptome and you only have a specific group of genes, for example, just 500 genes, or 300 genes, that I will probably avoid the highly variable features, because if not, you are going to remove a lot of your RNA information. Then we are calculating the PCA based on the highly variable features for RNA. So if we print again the Visium object to see what information we have right now. So a new slot has been added now here. So we can observe that for the RNA, we have PCA calculation, but we don't have any additional steps regarding the dimension reduction for protein yet. Now I'm going to calculate the UMAP. All of these functions in Giotto have the default feature type as the RNA. So that's why I'm not specifying right now that I want to work with RNA, because it's part of the default values. But you will observe in the protein section that in that case, in the protein case, we need to specify the feature type. So for RNA, you don't need to do it. But if you want to specify just to make more clear your pipeline, you can do it, of course. You just need to add the argument fit type, fit_type, which is indicated the feature type name, and you have to type RNA there. Okay, so we have the nearest network now. We are going to perform the clustering. So in this case, I'm using the resolution equals to 1, but of course, you can modify this resolution for your data set. There it is. Okay, so now we are going to plot the UMAP. Now remember that this UMAP is only considering RNA information. So we don't have the integration yet, we don't have proteins yet. This is only coming from RNA. So we have very well separated clusters, as you can observe here. We have the different colors from the gradient clustering, and we also have a good number of clusters using the resolution equals to 1, so we don't need to lower that number. We can also observe the distribution of these latent clusters in our sample. So we can observe that since we are plotting also the image in the background, this organization of the latent clusters are matching actually well with the different structures in our tissue. We can make the comparison, but observing the tissue alone. So if we put here the tissue image, you can observe these structures in the tissue. And we are expecting, of course, that these are particular cell types. So we were expecting to see these circles in our clustering. So it's good to know that we are already observing these structures, but as you can see, they are not completely perfect. For example, some of them are not showing really the inside of the circles, And also some of them are missing part of the boundaries. For example, this one here, even if it's almost surrounded by the brown spots, we are missing a few of them. So it's good. It's not a bad result, but of course, if we add more information, we may get a best result than this. So let's see now how it looks like running the protein alone. So for the protein processing, basically we need to follow the same steps. We are going to do the filtering, but observe that in this case, I'm specifying here under feed type that I want to work with the protein. So I'm also changing a little bit the parameters because in this case, remember that we have a maximum of 35 proteins. So of course, we cannot expect that the number of features per cell is very high. So the only, let's say that the only filtering that I'm doing here is that I expect at least not have an empty cell, right? Cell with no proteins. So that's why I'm being very flexible here, and I'm only asking for at least one feature per cell. And then since I have a big data set, that's why I'm keeping the filtering argument of having one feature in at least 50 cells, because I want a good representation of the proteins in my data set. So in fact, with these parameters, I'm not removing any additional cell, and I'm not removing any of the proteins. So basically, the proteins are very well distributed in my data set. I have enough information in my data. And just to explain a little bit more, the number of cells that I'm starting with is the number of cells that I'm keeping after the RNA filtering. So this is not the starting number of cells from the dataset, from the whole dataset. I already performed a filtering using the RNA feature type. So I remove cells. If I'm removing cells that didn't accomplish the filtering from RNA, then I cannot use those cells for the protein. I need to keep only cells that have information both from RNA and protein. So just wanted to clarify that. So this filtering is being performed on top of the filtering of the RNA, okay? So after doing the filtering, I'm going to also perform the normalization. No worries about this warning. Yes, the normalization was originally designed for RNA, but it's okay. We can still apply it to proteins. It's just giving you the warning so that you are aware of that. And then I'm going to run the statistics. So here, observe that I'm also specifying the feature type, because basically I want to keep the counts of the number of features per cell using the RNA, but I also want to know how many features per cell in the context of the proteins I have. So those are two different statistics. And basically, I'm creating two different metadata tables, which will provide the information from one or the other modality. Now, moving to the dimension reduction. In this case, since I have a very small number of proteins, I don't want to calculate the highly variable features because I will keep a very, very small number of proteins. So I want to consider all the proteins that I have available in my data set to create a PCA. And now after the PCA, I'm going to calculate the UMAP. I'm going to calculate next the t-SNE. Then I want the nearest network. And finally, the latent clustering. Here again, I'm specifying the feature type as protein, and I'm using the resolution equals to 1. Okay, it's on. Now let's plot the UMAP. Okay, so this UMAP is only considering proteins, and also the latent clustering is only considering the protein information. So we can again observe that the clusters are very well separated, and the number of clusters seems to be good. I'm plotting here also the network, but remember that you can hide the network by setting this argument equals to false if you only want to see the UMAP distribution, but not the connection of the cells. Now I'm going to plot the spatial distribution of these clusters. So again, this is only considering the protein. So we can observe again that we are capturing the tissue structure here with these circle areas. But again, even if they seem to be detected, we are still missing, for example, some information about the boundaries of these circles. We were able to capture, for example, for this circle over here, the inner section, which we were not able to capture using the RNA, if you remember that plot. So for this circle, we were able to capture in the RNA, the circle structure and the boundary, but not the inner section. Now with the protein, we can capture the inner section, but not very well the boundary. The same thing over here, right? So, okay, we know that the individual modalities are giving us good information. So they are not completely bad results. They are able to resemble some structure of tissue. But let's see if integrating the information of RNA and protein, we can get a better result. So for being able to calculate and integrate the modalities, we need to perform the k-nearest network. If you remember well, in the nearest networks calculation, the default is calculating the shared nearest network, the SNN, and not the KNN. So we need to calculate the k-nearest neighbors network for each modality. So we need to do this for the RNA first, and then for the protein. If you calculated the KNN instead of the SNN previously for the latent clustering, then you don't need to perform this step again. And then I will start running this step while I do the explanation because it's going to take some time. We have integrated in Giotto the weighted nearest network analysis. This is not something that we developed. This is a very well method developed in the past, and it has been also included in Seurat for performing this multi-omics integration. So basically, the idea of this analysis is that considering the dimension reduction results, so we need to start from the PCA results of the individual modalities, then you try to understand how much contribution or how much information is given the RNA and how much information is given the protein for describing the behavior of each spot. So basically, you get the weight of each of the RNA and the protein for each spot, and with that, you basically complete that from the 100% of the behavior of each spot, how much, let's say 30% is being described by the RNA, and then the 70% is described by the protein. So you are going to weight basically the dimension reduction behavior based on those proportions. And with that, you are basically integrating the information coming from the RNA and protein. In the future, we are planning to integrate also epigenetics information. This is not yet ready, but we are working on that. So we hope to include the integration of epigenetics, either coming from a TAC-seq or from the measurements of certain histones for performing this same step. So the method will be the same for now. In the future, probably we will integrate more methods for the multi-omics integration. But for now, when we integrate the epigenetics step, we are going to use the same WNN. And the only difference is that you will have to specify here that you are using, instead of protein, maybe a TAC-seq or something. So, okay, once that you have performed the WNN, this function is basically just printing the different steps. At some point, it needs to calculate some distances from the RNA, and then some distances using the protein. Then it's calculating the weights here, and then it performs a normalization, and finally, it will save the results in the same Giotto object. So you can see the results using the Get Multi-omics from the Visium, and then Well, let me see what I'm missing. This has to be the theta weighted matrix. I'm not able to find it right now, but let me see. Okay, here it is. I'm just not calling it for the right name. So basically, this is a super big matrix. Probably I should just bring the first 3 rows and columns. So basically, this result is it's a big matrix showing the comparison between each cell with the other one. So it's being cell to cell, and it's providing the matrix for the weights. Sorry, not for the weights, for the distance between each of these cells. So I believe this is this must be a different measurement. I'm not giving the right explanation here, because if you compare one cell to the other one, you have a similarity of 1. So I think is showing how similar they are between each cell. And then you have the weights for each modality. So this matrix is not exactly the weights, but you also have the weights here. The weights for the RNA, and basically here is showing for each cell or spot how what's the weight of the RNA, and then for the protein is showing basically the same. So for each spot is showing you the weight for the protein. Now, considering those results, we can calculate again the UMAP. So in this case, it's going to use the weights that we have calculated, and also the dimension reduction and the nearest networks for the individual modalities. But it's going to weight that using the weights that we calculated in the previous step. So it's calculating again the nearest neighbors, but based on the integrated results. Let's wait a little bit for this step. Okay, so after calculating the integrated neighbors, it's calculating the graph, basically to calculate the distances between the spots. Then it's calculating the UMAP, the integrated UMAP. And now we are going to calculate the latent cluster, but this time using the integrated k-nearest networks that we calculated in the previous step. So these integrated K and N are coming from this step, from the integrated UMAP. So inside is running the calculation of that k-nearest network. And then we are going to use that one for calculating the integrated latent clusters. Okay, so it's done. Now we are going to plot the UMAP, but this is the integrated UMAP. Here you can observe that the dimension reduction name is the integrated UMAP, and then I'm going to color the spots using the integrated clusters. So this is the result. In this one, the result is already considering both of the omics. So we have again a good separation of the clusters over here. We have a good number of clusters in using this resolution. We have a cluster here completely away from the rest of the cells. And then let's observe the spatial distribution of these integrated clusters. So here in this result, we can observe that we are getting a better description of the tissue because, for example, in this circle over here, we are now able to capture pretty much the whole boundary, but also the intersection of the circle. Then for this one over here, in the past, we were not able to capture the boundary and the inner side. We were able to just capture one or the other. So now with the information of the both modalities, we can capture both the boundaries and the intersection. And the other ones are pretty much in the same situation. They have both sections in the tissue. We are still able to capture other cells here, for example, in the boundaries. So as you can observe here, we have basically we can get a better description on the structure of the tissue when we consider both modalities. So that's basically the end of this section. Are there any questions about the integration? Oh, I see a question in the Q&A. So yeah, I think at the end I showed that the latent cluster is being performed using both of the RNA and protein information. So it's been an integrated latent clustering. Probably it's been confusing because I put here, when calculating the latent clustering, I'm saying, sorry, here, I'm saying feature type equals to RNA, but this is only right now the place to store the results. I think I will try to, in the future, update this tutorial so that I can create basically another feature type in the Giotto object, showing the combination of the modalities. Right now, I'm just putting feature type for storing somewhere the results of the latent clustering, but the actual calculation of the latent clusters are integrating both modalities. So no worries about that. This is only for finding a place where to save the result.